Okay, everybody, welcome back. If you're going to go feral this afternoon, can you please do it politely? And if you need to go feral to stay awake after lunch, that's okay. But for want of a better word, what we're about to get into really is the dark science of what's happening in the aquifer. And it is, and I literally say that, because if you drive around at things on the surface, in my job, if somebody sees a pothole in the road, they tell me. But under the ground, we can't see what's going on. So we have to use the best of science to understand it. And what you're about to hear from are the best science brains we have in New Zealand, and arguably some of them internationally, who have been looking at the Heretaunga aquifer and the plains for some considerable time. So we're drilling down now into what is going on, how the aquifer works, and what are some of the challenges. So we have four highly qualified speakers this afternoon. Dr. John Quinn, the NIWA Chief Scientist, Dr. Andy Hicks, the Principal Scientist for Quality and Ecology for the Hawke's Bay Regional Council, Dr. Brett Gilpin, the Science Leader of ESR, Microbiological Risks, and Murray Close, the Principal Scientist for ESR. And then after afternoon tea, we're going to get another science presentation. Now, it might sound a bit dry, but trust me, it's not. And this is actually where the rubber meets the road. This is where you'll see what's happening, meets the road, should I say. This is where you'll see what's happening. So we're a bit ahead of time, and I've deliberately done that, because I think actually you need some time for some questions, probably more than we might have in the other sessions. So without further ado, it's my pleasure to invite Dr. John Quinn, the Chief Scientist for NIWA, to present to us.
Kia ora koutou katoa. It's a real pleasure to be here in Hawke's Bay. I actually come from Waipukurau. I was born in Takapau and spent a lot of my university days working at Whakatū and enjoying the waves at Hamoana and Tiawanga. So it's lovely to be back here and stay with my good friend Richard Lee last night and catch up with people. So I'm going to talk to you a bit of a high level talk about what's going on with fresh waters, surface waters in particular in New Zealand. So first of all, I just want to start off with thinking about, when we talk about our fresh waters, we start off with our groundwaters. We've got 600 billion cubic metres of groundwaters in New Zealand. Those groundwaters feed into our wetlands. Now, our wetlands are greatly diminished. We've drained nearly 90% of our wetlands over the last 150 years for farms and for towns, but those then feed into our streams. We have an amazing diversity of streams, which I've spent my career working on, having got hooked on rivers whilst trying to catch fish in the tuki as a kid. And then we've got a wide variety of lakes and 300 estuaries. So, we have an amazing resource of fresh waters in New Zealand. For Maori, freshwaters have always been really important to them culturally and as a food resource and as a resource, and also they have a spiritual relationship with water, which I think the rest of the Pākehā community are learning from. For a lot of Maori who have been disenfranchised from their land over the last 150 years, being able to continue to interact with fresh water has been a really important part of maintaining their culture. And Sir Tipiti O'Regan gave a talk at the Freshwater Sciences meeting last year and talked about freshwaters as the hinge of cultural heritage for Maori. So we can't really underestimate the value that freshwaters have to Maori. Freshwaters are also a really important economic resource for all of us. They provide the hydropower, which gives us clean energy. Most of our electricity comes from hydropower. Water for irrigation for our farms, water that we drink, water for our industries. Also other resources like gravel that come out of our rivers for our roads, for our buildings. But we use waters for recreation, for our fun times, for our tourism, and waters are also receiving waters for drainage waters and effluents and runoff as well. So they really are at the heart of a whole lot of conflicting values, but really important to us as New Zealanders. The need for freshwater restoration has become very clear over the last decade, and a lot of it has been crystallised in a couple of reports that have just come out in the last couple of months here from Sir Peter Gluckman and from the Ministry for the Environment and MPI. really stating that we have a lot to do to actually improve our freshwaters to levels that people are happy with to provide our needs. So I'll just go through some of the recent, we look at the current state and trends across our fresh waters. There was a review that one of my colleagues, Scott Larner, did just looking at the last 10 years of data from the Lawa data site that Liz talked about earlier. So data is collected by NIWA and by the regional councils, around 1,000 sites from the period of 2004 to 2013, and looking at what's the state and trends of those waters. And if we look at some of these indicators, and they've been divided up by the dominant land use in different catchments as a way of looking at those, dividing up that broad range of data. And what we see is we go from native land cover, which might be forest or tussock, through to production forestry, to pastoral agriculture, to urban. There's a general degradation of ecological health, so we use this Macroinvertebrate community index as one way of looking how healthy our rivers by asking the insects that live in the stream beds. And we go from excellent health in the native cover through to pretty poor, on the margin of poor to dodgy to pretty bad in urban areas. And if we look at the nutrients, nitrogen and phosphorus, now we're concerned about those because they grow the algae that you can see on the slide there in our rivers. When we get too much, like a lot of the things that Lou was talking about earlier, we need a certain amount of nitrogen and phosphorus in our systems to make them productive. But we get too much, we get all sorts of problems with overproduction of slimes and things that flow on from that. So we see increasing levels of nitrogen and phosphorus as we go across this gradient from natural cover to urban. And reduction in water clarity, as we go and looking across the bottom line of things to do, particularly with swimming and swimmability. Our streams and pastures in particular tend to be dirtier than those in other land use types. And in terms of meeting the E. coli grades that are set for safe to swim, and there's a couple of different lines there. There's the red line at 130 per 100 mils is the new clean water policy proposed as a median that sites should have to meet. And a lot of our pastoral sites and urban sites are not meeting that, and nor are they meeting the 95th percentile, which is trying to keep the really high levels down. So we've got work to do there in terms of state and trends, particularly in our pastoral environments and urban. But if we look at what's been going on, so this is the state, but as Liz said, the trends are really important. What these graphs show here is of those 1,000 sites, there are about 500 for which there were enough data to look at what's the trend, what's been happening over that 10-year period. And we see that a number of these things that are on the left and in the middle there, so what this is showing is the proportion of sites that had significantly degrading water quality over that period, which is in the orange bars, and the blue bars are those that were significantly improving. And you can see that a number of things, the clarity, ammonia, E. coli, and dissolved reactive phosphorus, we're actually seeing more sites that are getting better than getting worse. And those are mostly things that are controlled by things we do. They get into water and surface runoff processes. So the work that's been done with fencing livestock out of streams, better effluent management on dairy farms, there is some evidence from here that that's been having some benefits. However, on the other side of the graph, the nitrate, which tends to come through the groundwater and has a longer lag time before it comes to the surface waters, we've got more sites getting worse than getting better in that area. And the macroinvertebrates, there's not so many sites to do the trend analysis on, but again, we've got more sites getting worse than getting better for macroinvertebrates. And there are some issues around lag times for insects being able to get to places where the habitat's been restored that may be part of that story. So the state and trends. I just want to tell you a little bit about a tool that's just been released by NIWA in the last month called New Zealand River Maps. And a lot of the information we have in the Lawa database and the stuff I've just shown you is for a whole lot of points around catchments. But people are often interested in what's happening at their place. So we've developed over the last 10 years or so a number of models that let us predict what the water quality will be at all through catchments using statistical models. So this New Zealand River Maps has been developed, and there's about 101 variables under these different topics, which we can now predict through the whole river environment classification network of New Zealand what we're expecting to see there. It's not the truth, but it's what we predict to help you explore what might be going on. So there's a little website there. It's a shiny app, one of these clever things that people do in our computer system these days. And I'll just give you a few examples of some of the things that can do for the Ngaruroro, the Tutaikuri, and the Karamu. So the first one here is for total nitrogen, and you can see I've selected the slider here as for third order and greater streams, and as we go from blue to low levels of total nitrogen through to orange, we're getting increasing levels. So you can see immediately from this graph that the high levels of total nitrogen is a median, particularly in the Karamu and the lower parts of these catchments. It's quite a similar story for phosphorus, but some higher levels of phosphorus in the Tutaikuri as well. This one here is for dissolved oxygen, so it's predicting. So there's a lot of macrophyte grows in the lowland streams of the Karamu, and that results in pretty low oxygen levels at times, but pretty good in the other river systems. This one is for E. coli. Remembering that the median for E. coli, if it meet the bathing standard, is 130, so you can see there's a lot of sites there in the Karamu that are above that. They're going to have to be dealt with. And the last one here is looking at this Macroinvertebrate community index, looking at the various levels, and the ones that are around, the dark oranges are getting into the bad ecological health state. So there's a whole lot of information about predicting fish, predicting, actually this last one here is on the predicted maximum water take. So it gives predictions if all of the consented water takes were taken at the same time, which doesn't happen, this is how much water take cumulatively there would be down through catchments. And you can go into this Explorer and look at surface water takes and groundwater takes and takes for different uses. So this is a pretty cool resource that you can access for free to explore anywhere in New Zealand. Moving on to lakes then. So we've got nearly 4,000 lakes greater than a hectare in New Zealand, but we only have really data for the water quality on less than 5%. And the data set tends to be a bit skewed to places where we've got problems, so it's not truly representative of New Zealand, but nonetheless, this is what it's showing us. We measure lakes by their trophic status, which is a low level, a microtrophic or oligotrophic is low levels of nutrients, low levels of chlorophyll in the water column, good water quality, water clarity. And you can see we've got nearly 40–50% of the monitored lakes are in the eutrophic and higher levels. So we've got some real problems with our lakes, and this is the sort of thing that you get when you're into eutrophic conditions, these sorts of scums along the side. And people who've been to Tera will have seen this at times in your problem case in this area here. This is just a little bit of an advertisement for a book that we're just in the process of finishing on, a lake restoration handbook. It's been put together by Niwa and colleagues at the University of Waikato. I guess I wanted to put this up to show a couple of things. This is a complicated system, a lake, and if we're thinking about restoring them, we need to think about restoring the inputs. We think about pest fish, pest plants. It's quite a complex system, and what this book tries to do is to put all that together. And some of the interventions involve doing things inside the lake using flocculants, but any lake you're trying to restore, you need to think about it very holistically, and that's what this book is intending to do. That'll be coming out hopefully within the next 6 months. It's really just a matter of getting it through the publishers now. So what are the big picture key challenges for us, for our fresh waters, surface waters in New Zealand? The first one is around habitat loss, and often the debate is about water quality and contaminants, but really habitat is very important for our native species. And the drainage and flood control works we put in place have quite a major impact. If you think about the floodgates, that's the one shown there, flood and tide gates, they restrict a lot of, most of our native fish spend part of their time in the ocean and part of it in the fresh water. And it's been calculated in the Waikato, there's about 25% of the available habitat for inanga they can't get to because of floodgates and tide gates. So that has a big impact. With water abstraction, we're hearing about that, reducing flows. As I mentioned before, we've drained a lot of our wetlands. A lot of our streams have had all their riparian vegetation taken away from them. That has a big impact on them. We've got invasive plants and fish like koi carp. Not so many of those down here, but in the Waikato, these are a real problem, contributing to the degradation of our lakes. And then we've got the contaminants that you've been hearing about, and including legacies, particularly in the things that get into groundwater systems, the nitrates. And on top of that, we've got climate change coming. So we've got a fair old range of challenges to deal with. But there are a lot of tools in the toolbox, and I've just put a few examples up here for things that we've been working on at NIWA and a lot of other scientists in CRIs and regional councils and universities. Things to do with capturing the nutrients and energy that comes out of wastewaters, turning that into products, different ways of getting fish through things that are barriers, big long culverts which otherwise they couldn't have swum through, putting baffles to get them through, geotechnical ways of flocculating out and capping phosphorus that's in lakes, treatment wetlands, artificial wetlands, put them on the end of tile drains, and repairing buffers. We know all of these things are really important tools, and there's a whole lot more being developed as well. Just want to move on to a little bit about water availability. New Zealand is really blessed with a lot of fresh water compared with internationally, but that water, as was mentioned earlier, it's not uniformly distributed around the country. It's often not in the right place or at the right time. And if you look at the graph here of the 7-day mean low flows, you can see the west coast got lots of water, but the east coast of the North and South Island and Northland are much lower water supplies, and you all know that. And by the time we take allowance for the water that the environment needs, the amount of water that can be allocated, in many cases it's near or at or over the allocation limits. So we have to be much more frugal with how we use our water and use it more efficiently. Looking at future challenges in that area, this graph shows predictions for, compared with 1990 through to 2090, with climate change, what we're expecting to see in reductions in mean flow around New Zealand. And it's a bit of a case of the rich get richer and the already water short get poorer. And so the places that are in orange, we're expecting between a 5 and 20% reduction in mean flow by 2090, and the red ones are above that. And you'll note that the Karamu, parts of the Tuki catchments are in those places. So we've got things to deal with into the future. We're also expecting to see increased storminess, and from some of the weather events we've had in the last month, we know what that does, particularly when it's linked to sea level rise. So we're going to have some real hazard issues to deal with in our lowlands and much of New Zealand. We're expecting to see increasing populations, so there'll be more people around, and more irrigation as people try to feed the planet and the people in New Zealand. So these, combined with our cumulative effects of takes and ongoing use of fertilisers, and we have a lot of things that are accumulating and legacies. So these are all big challenges for us to deal with. And if that wasn't enough, with more people coming to New Zealand, greater ability to buy exotic plants and species over the internet and smuggle them into the country, the biosecurity stuff is not going to go away either. So we've got some big things to deal with. However, just with the land use issue, a couple of projects I've been personally involved with over the last 15 years or so. One at Whatafoda, a hill country farm between Hamilton and Raglan, where we worked with a group of farmers in the late 1990s on a farm that was not making enough money and was producing a lot of contaminants. And we looked at, well, how can we make this whole system more sustainable economically and environmentally? And we looked at land use capability, what was being done now. We found that half the farm was actually losing money every year because of the amount they were spending on gorse control and fixing the fences every time a slip went through them. So that land went into growing pine trees. That really increased the overall profitability of this land, but it needed, there was an investment hurdle to get into that new system. And that was dealt with by bringing in some external funding. What that's done is improve the overall sustainability of this land. The habitat's improved greatly. Some of the issues around nitrogen haven't been cracked. There's some moving baselines associated with that, but also by shading out a lot of the streams, we've reduced the nutrient uptake in the plants in the summertime, so we're getting more nitrogen coming out in summer. So it's difficult to win on all of the swings and roundabouts at once. The other one is Te Awa Waitawa, working with iwi around the harbour there, who wanted to fix the harbour. They came to NIWA saying, well, how can we fix the harbour? We need to look to the catchment was the answer, so I got involved. They did some spearheading work with dealing with their own farms and then talking to the rest of the catchment. The other farmers in the catchment got on board. They've set up their own nursery. Over 10 years, they've fenced and planted 80% of the streams in that catchment, and they've seen reductions in nutrients and improvements in stream health. And they all won a community award at the last Rivers Foundation Awards So, when people get together, New Zealanders can crack these problems. We have to scale up, but we've got the talent and we've got a lot of the knowledge to do that. So, well, I'm finished. So, conclusions. We've got very valuable freshwaters. They really are under increasing pressure, though. The things that have been going on around setting limits and restoration, we are getting some things to make people think a lot. We put the necessity there, will be the mother of invention. And some things improving, others aren't, but we're all in this together to try and use the tools in the toolbox to crack the problem. So thank you for listening.
Thank you, and I do apologise because I got it round the wrong way. We're doing above surface stuff first, and after afternoon tea we're doing the dark art underground. So you guys know what's above the surface. So now it's my pleasure to invite Dr. Andy Hicks from the Hawke's Bay Regional Council.
Tēnā koutou katoa. Andy Hicks from the Hawke's Bay Regional Council. I'm the Principal Scientist and Team Leader for the Water Quality and Ecology team there. I'm actually from Australia, a lot less local than John, even though I live and work here now. But I make no apologies for that, for being Australian, that is. And I hope you don't hold it against me. But yeah, so today I'm going to John's talked about the bigger picture, the national. I'm going to try and focus in on what's going on locally. And John also talked about some modelling exercises, which we can compare with the results that we've been collecting. And they say the same thing, which is always nice. Sometimes models tell you a different version of reality, so it's nice when they do match up. Okay, yeah, so before we get into it, I'm going to show a series of slides that have a similar setup to what's here. So on the left, you've got the Tudaikuri, on the left, you've got the Tudaikuri and Ngunnawara rivers, and then on the right, you've got the Karamu catchment. The blue circle is the Heratonga aquifer. So a lot of the talks today are concentrating on that zone, but for the surface water and for the aquifer, which Jeff will go into a bit later, it's always important to think about the headwaters all the way out to the sea. It's all connected and it's all important. And on that topic of interconnectedness, it's also very important to think about land and water at the same time. And so that's what these slides are doing. The green is where you've got forests, either native or plantation. The orange is where you're starting to get into cleared areas used mainly for grazing or other forms of stock production. And then when you're getting into the purple and the red, you're getting into cropping and more intensive forms of vineyards and orchards and these kinds of things. And then the grey areas are urban. So what should stand out from these first two plots is in the Tudaikuri Ngunnawara, you've got a large area of the catchment in the headwaters where it's relatively pristine coming out of the forest, and so that influences the water quality that you see even down near the coast. And on the right, the Karamu, the water starts in areas that are already heavily modified, and that has a real influence on the water quality patterns that we see. I should also mention you won't be able to see a lot of the details in these maps, which is okay. We're just looking at the bigger picture. The reports are all available online. So these are Hawke's Bay SOE type reports, which are actually authored by Sandy Heidecker, who I think is in the audience, if you just wave out. I recognise a few of you guys from the tank stakeholder group, so you'll recognise Sandy more than you do me. She's our lead scientist for the tank area. So that's the land, and now we can start to look at the water quality patterns. So all of these next plots will have dots on a map, and those dots tell you how much of whatever we're looking at there was, and the colour indicates whether it's good or bad. So blue is good, red is bad, small is small, big is big. And so this is E. coli, faecal contamination. A lot of people will know all about that after the Havelock North incident. And this is the thing which really influences whether you can consider it safe or not to go swimming. This might not work, I'll try it. Can you still hear me? Not so much? No. You can? Yeah. Okay, good. So up here, the dots, you might not even be able to see them. They're so small and they're blue. As you work down into the catchment, you start to see a few higher levels. But in general, in this catchment, it's relatively good. Low. And that's because The water coming out of here, there's a large volume of that, and even as you start to get a little bit of input from the tributaries down in these lower bits, that stays relatively low concentration because it gets diluted. And that story is consistent with a lot of the other variables that we're going to look at. By contrast, in the Karamu, you can see that the numbers are relatively high. They're in the darker colours. Going back to what John was talking about, the yellow is where you'd want to be considered safe to swim according to the new rules. And you can see a lot of the areas of the Karamu are at or above that limit. So that's E. coli, faecal contamination. Then you've got dissolved inorganic nitrogen, or DIN. Nutrients are really important for farming, so a lot get put on. But those nutrients also find their way to waterways, and just as it makes plants grow really quickly and productively on land, and makes the algae and sometimes nuisance plants grow really quickly in the waterways. So nutrients are important, and this is nitrogen, which is one of the main ones we need to worry about. So same kind of picture. In the Nararoro Turaikuri, relatively good levels throughout. In the Karamu, you're starting to get some higher levels. And then contrast that with phosphorus. So if you're a farmer, you'll know phosphorus and nitrogen, the two big ones. With phosphorus, we tend to see, in general, higher levels across the region and also in these catchments. And then by the time you get to the lower areas, and particularly in the Karamu, this one again, quite high levels. So there's plenty of nutrients in the Karamu system. By the time you got down to the bottom of the Turaikuri and Nararoro, you're also seeing quite a bit, particularly in the tributaries, less so in the main stem. And then Periphyton slime algae, which you can see there. So the one interesting thing about Hawke's Bay, interesting or problematic, it's a headache, is that even though the nutrient levels aren't particularly high, we still actually have periods of time when we have very high levels of slime algae in the rivers. And you'll all recognise that when you drive over or go fishing or go for a swim, and the bed of the river is covered with bright green stuff or brown stuff or sometimes black stuff. And that we see up the main stems of both rivers, in the tributaries, and also in parts of the Karamu. At this point, I should say that there's two really main types of waterways. Hard bottom streams are the ones with rocks and cobbles on the bottom, and that's what slime algae and Periphyton attach to. In the Karamu, many of the streams are another type. They're sort of more soft bottom. These are the areas where you get aquatic plants rather than algae per se. And so we don't see the slime algae in those areas because there's no suitable conditions for them to grow. And so we look at macrophytes in those other areas, and I'll come back to that a little bit later on. But yeah, so we've got problems with slime algae in Hawke's Bay, and that's going to continue to be a challenge because nutrients are part of the problem. They certainly need nutrients to grow. When you've got a lot of nutrients, they grow faster. But in Hawke's Bay, when you've got low flows and really hot weather, the algae has a long time to grow, and that means it can get to quite high levels, even though the nutrient levels aren't as high as you would usually be worried about. So we'll continue to have problems with slime algae even after we've got our nutrient levels down, and that's something we need to think about. And the last contaminant I was going to talk about was sediment. And so what we've got on the plot here is turbidity. Same kind of pattern, low in the headwaters, gets worse as you go downstream. But the difference is in the Karamu, you don't tend to have high turbidity because it's one of these more lowland systems, very flat. So the sediment doesn't get carried by the water as easily, it settles out. And that's why they're soft bottom streams, because the sediment all settles out rather than being moved through quickly. And so the turbidity levels in the Karamu are actually quite good. When you look at how much sediment is getting to the coast, not that much is coming from the Karamu, whereas a lot of it is actually coming from the Turaikuri and Nararoro. So a different kind of pattern. And it's important to think about where all that sediment's going, because although our turbidity is quite low in the rivers, the waterways, there's still a lot of it coming out. And in that case, it's really having a large impact on the estuaries and on the coast. So we don't stop when the fresh water becomes salt. We've got to think about what it does as you get all the way out to sea. And in our estuaries in particular, we're starting to see an increase in fine sediment, which is a bad thing for the health of those catchments, or those ecosystems, those habitats. But I'll leave it there because we're not really talking about that stuff today. And so, yeah, lastly, macroinvertebrates are bugs that live in the stream. These are the things that we use to give us an indication of the overall health of the waterways. So some bugs are very sensitive to pollution, to warm water, to nutrients, to things like that. And so when you look at those bugs, they're experiencing the water for a long period of time. So although you might miss pulses of contamination or pollutants that move through the system, those bugs were there when it happened, and so that if they're dead and they don't recover, it gives you a better long-term picture of what's been going on. So they're a really good integrated picture of the health of waterways, and so we tend to use that as our overall indicator of ecosystem health. And this probably shows the strongest pattern that we've been seeing today in terms of the headwaters, really good. By the time you get lower down, things aren't looking as good. And in the Karamu in particular, we've got lots of red blobs. I changed the size of these blobs so it looks a bit different, just so it's clear what I'm talking about there. And I'm going to talk about that a little bit later on. But a lot of people ask, is the water quality good or bad? And that's a really hard question to answer because water quality covers such a large range of things. So it's really hard to say whether it's good or bad. You often need to get a bit more specific. Are the nutrients high? Is the sediment high? That kind of thing. But Sandy's done a really good job trying to collate all of this information. And you won't be able to see the details, but I'll just talk through them and look away from you while I'm doing that. So we've got the Nararoro here. We've got the Tutaikuri here, and we've got the Karamu here. So each row is a site in those larger catchments. And then the columns are those different variables that we look at. So there are some things that we think about when we're grading against toxicity. It's looking pretty good just about everywhere. Algae and weed growth, looking okay, except when you get down to the Karamu. Nutrients, the nitrogen and the phosphorus. That picture where phosphorus tends to be a little bit worse than the nitrogen, you can see here, and you can also see that the Karamu again stands out as being particularly bad. So incidentally, that's the waterways around where we're sitting now. We're sort of in the Karamu catchment now. Sediment is not looking particularly flash in terms of water clarity. Turbidity is not too bad, but yeah, so that's a bit of an in-betweener. And then our indication of overall ecosystem health, the bugs that I talked about just before, you see that same pattern. So good in the headwaters, as you get down, not so good. And so this table, you can kind of see where does all the red blobs concentrate. We're really talking the Karamu, and so I'd like to explain what's going on there. So yeah, why is ecosystem health so poor in the Karamu? And so again, Sandy did a really neat investigation trying to get to the bottom of this, going out, looking at nutrient levels, looking at toxicants, looking at the habitat quality. And the two things that really stood out as explaining what was going on was oxygen and temperature. I'll turn away from you again, sorry. So these are the kind of bugs that are really sensitive to pollution. So if you see them in the stream, it's good. And these are the ones that are less sensitive, so more tolerant. The red is the sites that scored poorly. When you look at these bugs, we didn't have anywhere that scored excellent. And then the good and the fair are only a couple of sites. So there are a few of these guys here, but mostly these guys were everywhere. And when you look at the oxygen, you can see where we had the best score, oxygen levels stayed high, and the temperature never got too warm. So the red is getting hot, the blue is staying cool. These black bars, if you're on this side, that's bad. If you're on that side, it's good. And when oxygen levels stayed high throughout, that means the bugs were never exposed to oxygen stress. And when the water also stayed relatively cool, they were never exposed to that heat stress. So not surprisingly, systems where you had nice oxygenated water where it never got too hot is where you get the good bugs. And there weren't very many of those places in the Karamu catchment. And so just to explain, the oxygen side of things. So when you do have these open drains where you get lots of sunlight, it's easy to imagine that it's going to get warm because that sun cooks up the water. But what's less obvious is that when these weeds grow in the stream, they're often called oxygen weeds, and that's true. Plants produce oxygen during the day when they're doing their thing. But at night time, they behave like any other thing. So there's no sun, so they're not doing their photosynthesis bit and generating oxygen. They're just consuming oxygen. And so when you've got a really large biomass of anything, including the weeds, they use up all the oxygen that's in the stream overnight. And so that's what drives the really low oxygen levels. And this is a major factor of why these streams don't have good ecosystem health. So how can you avoid that? How can you make things better? And it really comes back to riparian management. So this is a relatively pristine looking area. It doesn't need to look exactly like that, but the idea is you plant trees, you plant tall vegetation which casts shade, that intercepts the sun, and that means that the weeds don't grow as much, which looks after the oxygen levels. And it also creates a lot of good habitat by limbs and leaves and things falling into the stream, which different kinds of critters like to live in or eat. And it also creates your terrestrial biodiversity, so you've got all sorts of benefits when you've got a nice riparian zone. And, couple of minutes, perfect, thanks. And so, yeah, obviously we can't get every little drain, every little waterway looking like that last picture, but this is a little experiment that we ran not too far from here, out off Mutiny Road. So there was vegetation all the way along here. We went, it was an introduced pest, so we didn't worry about cutting it down. So then we cut it down here to see what would happen when you had good shade versus no shade. And hopefully you can all see a very strong difference here and a very well delineated line. And we didn't get in there with the digger and make that happen. This is just how the weeds grew. So when you've got a drain like this, because you want the water to flow, that creates a big problem when you're trying to keep your paddocks in optimum agronomic type conditions. So that weed causes a problem for the farmland as well. So by shading it out, it means you don't have to get your diggers out there, it means you don't have to use a lot of herbicides, it means you don't have to continually try to maintain things because the shade is doing that for you. So the shade stops the weed growing in the first place and it saves a bit of headache. So we're doing a bunch of experiments like this, just trying to work out how we can best go about generating that shade, because it's expensive to plant trees and look after them. And you don't always have a lot of space. Sometimes people are a bit, concerned about giving up large areas of land alongside each of their waterways if they've got several kilometres of little waterways intersecting their property. So we're trying to work out ways of going about doing this and helping ecosystem health in areas where you've got that conflict between what's currently happening and what we're seeing in the stream. I'm pretty sure that's it. Yeah. So that was, thank you. Questions at the end.
Well done. Now we move to Dr. Brent Gilpin, who's the science lead of ESR. Welcome.
Thank you.
You want the clicker?
Yes, please. Kia ora koutou katoa. Thank you for the opportunity to take part in this meeting and talk to you about microorganisms. So all of the things on this slide are microorganisms, but the ones I'm going to really focus on are the bacteria, the protozoa and the viruses. Things that are generally around or less than 1 in 1,000th of a millimetre in size, so very small things. In the next 15 minutes covering these in any detail as possible, but I'm going to touch on pathogens, some of the sources of them, drinking water, look at a few outbreaks and some groundwater related aspects. So before I get into the bad aspects of microbes, let me point out that most microbes are actually are good. And you and I, for every 1 human cell we have, there are 10 microbes living either in or on our bodies. So we're mostly microbes. And these are mostly doing things to help us with our digestion, to keep us healthy and to do other things. So microbes are ubiquitous, they're everywhere, and generally they're good. But there are, of course, pathogens which are the ones that come to attention. As I said, viruses such as norovirus, the bacteria and protozoa, which I'll talk about in a bit more detail. And most of these guys cause some sort of gastrointestinal illness. So they're spread by the fecal oral route, and when you ingest them, a range of diarrhoea and other symptoms can occur. So So New Zealand has, this is a table of notified enteric diseases, microbially caused ones. So Campylobacter typically has 7,500, or last year there were 7,500 notified cases, of which 1,000 were from August last year. That outbreak here was in fact 5,500, but only 1,000 became notified, those that are actually met the clinical definition and were notified as part of the system. So, but the Hawke's Bay also has elevated levels of Cryptosporidium and Giardia compared to the rest of the country when you look at rates. And the ones in green are ones where people typically report recreational water contact more frequently than others. So New Zealand has a relatively high level of enteric microbial disease. So Campylobacter, which most of you in the audience are familiar with in one form or another, causes illness 1 to 10 days after ingestion, a whole range of symptoms, including diarrhoea and abdominal pain, and for a small number of people, some ongoing symptoms such as reactive arthritis and Guillain-Barré syndrome. And obviously the causative organism in the outbreak last year. We were, however, fortunate that the next organism, E. coli 0157, wasn't the actual causative organism. This also causes diarrhoea in people, but in 10% of children who become ill, they develop hemolytic uremic syndrome, which is can lead to kidney failure, and in 1% of people, death. So if we'd had 5,500 people in this region sick with E. coli 0157, you could have expected to have 50 or more dead children as a result of that outbreak, and a number of others who would be on dialysis and other long-going steps. I should also point out that this E. coli 0157, the E. coli we monitor in waters and which has been referred to, is one of those commensal normal organisms, so which doesn't cause any harm. This is sort of a bad cousin of the E. coli family that's developed this special ability to cause illness and disease. So the water quality guidelines, the monitoring for water, is looking for an indicator organism that indicates the potential presence of pathogenic organisms and really is an indicator of fecal contamination. So this is a particularly nasty bug that we have in New Zealand and which we should be pleased we didn't have last year. The only other organism I'll spend any time on is Cryptosporidium, which is a protozoan organism. Again, causes diarrheal symptoms. Some of the features of this organism are that it's resistant to chlorine treatment. So this, as part of its life cycle, forms these hard little spores that are quite resistant to a number of water treatment processes such as chlorination. They can also survive in the environment for months and are found quite ubiquitously in a wide range of sources. So where do we find these microorganisms, and how do they actually cause illness in people? Well, sick people, people who are ill, can excrete, in this case here, that number at the bottom, 10,000 million Cryptosporidium per day a sick person can excrete in their faeces. And you only need to ingest something like 10 of these to cause illness. So keeping human sewage, human waste, either from ill people or even people that are just carrying the organism, keeping that out of our drinking water supply has probably been one of the biggest public health measures of the last 100-odd years, and saves lives, a huge number of lives. And continuing to keep human sewage away from our drinking water and other sources is the biggest priority. But we also find these organisms in a whole range of other animals. So cows, sheep, domestic pets, wildfowl and other sources can all carry these pathogens in their system. The reason a number of these little animals here have smiles on their faces is because typically these organisms don't actually cause disease in a lot of these animals. They're part of their commensal microflora, just like the microbes on us. So they can have in their faeces, as you'll see, millions of these Campylobacter or other organisms per gram of their fecal material, which then can be excreted into the environment and cause disease. So each of these animals can have different levels of indicators and pathogens, and some of them may be less pathogenic. So for example, wildfowl such as ducks may carry Campylobacter, but they may be much less capable of causing disease in humans and others. So I'll just give you one bit of sort of data here from a study colleagues at ESR did looking at lamb faeces. So this is lambs excrete about 800 grams of faeces per day on average, a sheep about 1.5 kilos, and a cow about 25 kilos a day. So per gram of faeces, they can have up to 4.5 million grams of faeces, bacteria, Campylobacter per gram of faeces. So the entire outbreak last year, if it in fact went down through the well, head could have been caused from as little as 50 to 100 grams of faeces could have caused that entire outbreak. Also, in this case here, you'll see Cryptosporidium, Giardia, and these STECs, or E. coli 157s, carried in a small proportion of animals. And I could put similar data up for a number of other organisms, a number of other animals such as cows, deer, and other sources. So animal faeces does contain a considerable number of pathogens. So to keep those out of our water for drinking, whether it's from human sources or from these animal sources, a multi-barrier approach has really been adopted in New Zealand and is widely used, with five main components. Protection of the source water, so that's keeping that source water, whether it's surface water or groundwater, with as low a level of microorganisms as possible. Water treatment processes, which have two parts. The removing particles, which is around getting rid of those protozoa and large organisms, and then inactivation processes such as chlorination or UV to kill your bacteria or your viruses. Having a secure distribution system, then monitoring water quality, and then responding to any adverse test results quickly. So I'll just quickly, this is some data from some work by Dr. Steve Harudi, which is on the inquiry website. So these outbreaks worldwide, which I've just pulled out a few from this century, and so these ones are due to contamination of the surface water. So in these cases here, there's either a sewage discharge or either cattle or sheep upstream of the water intake for the drinking water supply. And in all these cases with the surface water, you really do have to do treatment. There were treatment failures in these cases, in some cases compounded by the amount of contamination that resulted in significant numbers of cases of outbreaks. Groundwater, which in many cases is either untreated or treated to a low level. There's a few examples of outbreaks causing a fairly large number of cases. Walkerton in Canada was previously the world's largest Campylobacteriosis outbreak, but also with some E. coli 157. So in this case here, contamination of the bore primarily in conjunction with rainfall events. There's also, you can have a really good source water, you can treat it well, but if you have your reticulation system not looked after well, these are examples where there's either cross-contamination occurring into the reticulation system, or animals, such as in the bottom case there, a dead rabbit that got a single rabbit getting into the part of the treatment process, that can result in some really large outbreaks occurring. New Zealand has had plenty of outbreaks, and this is just a selection here from the last 10 years, and in red, those involving Campylobacter. So the events in Havelock North are certainly not unprecedented. But of course, Havelock North is now the world's largest Campylobacteriosis outbreak, and so Havelock North has, well, New Zealand has even more reasons to be famous. So this is a quick shot, so I'll just quickly, here's the Brookvale Road bores 1 and 2, and the Mangatiri stream running along the background here. And these paddocks here, where several thousand sheep were being fattened up. So these sheep contained in their faeces this Campylobacter jejuni, and clearly of a virulent strain that also, as we do some more analysis, appears to be one able to travel through groundwater systems perhaps more rapidly and survive longer than previously thought. So there was an extreme weather event. A huge amount of rain occurred in the few days before the outbreak. That was, in this diagram here, shows thousands of cubic metres of water being washed off these paddocks, washing the sheep poo either directly towards these bores or into this Mangatiri stream. So effectively cleansing the poo from the sheep directly into the stream. This is the Mangatiri stream pond. So at the time of just before the outbreak, is really a dry stream, and the Campylobacter, which was resuspended off the paddocks, was washed into this pond and then able to be transported to the bores. So this diagram here, it's a little difficult to see. These are the Brookvale bores, so this is bore number 1, and which is sort of water being drawn from 10 to 20 metres below the surface. Across the top here is this impermeable layer or aquitard that really was thought was protecting this aquifer from surface contamination. But the Mangatiri stream here, you'll see, dips down almost through and potentially creates a pathway directly into that bore. So contamination was able to go in this event to those bores and then be sucked up and distributed to people. This afternoon you'll hear much more, well later on you'll hear much more about groundwater, but I'll just point out that this is sort of a typical diagram of groundwater, these sort of aquifers in big blue like an underground stream. That's not in fact how they look. This is what it actually looks like underground. So it's these sort of alluvial gravel aquifers with water permeating, travelling through them. So this creates an opportunity, these sort of gravel aquifers which are saturated, as the water moves through it, for microbes to be removed. And these are also the source of, these aren't just an inert, sterile environment, they're an active area of biodiversity. So this is, like Andy, an Australian organism in this case, this is a little stygofauna that lives below the surface in groundwater systems. This little guy here is actually only 1 millimetre in size, and those little yellow bits you see are either bits of sand or bits of bacteria that these guys are able to ingest, clean up the water in an aquifer. And these sort of organisms are just part of what is a whole ecosystem below the surface. So there's a whole lot of organisms, potentially hundreds or thousands of different types of these sort of stygofauna, but also large numbers of bacteria, fungi that all live in the groundwater system and can be quite effective at cleaning up the water through the groundwater. So as the water travels through soil, or through a groundwater system, you can inactivate or remove microorganisms. So filtration, physical removal, will take microbes out of the water. Predation by these sort of organisms, eating them, will actually remove them. Starvation, they like being inside our guts or an animal gut, so as the longer they're there, the better. So if you can increase the transport time that it takes from the surface to when it actually gets to your source of water, you'll increase the opportunity for these things to happen. Also in a surface environment, sunlight and desiccation can assist. And I guess the key thing with all water sources is avoiding direct deposition, because when faeces goes directly into any sort of water source, none of these processes are able, well, you get a limited ability of these processes. Yep. So
Okay.
a couple more.
Yeah, no, that's right. No, I wasn't going.
Yeah, that's right. So I guess if we look at the event last year, this multi-barrier protection of the source of the water was clearly wasn't occurring, and we've learned a huge amount since about the security of the aquifer, and the learnings from what's happened here are going to be transferred throughout New Zealand and indeed worldwide as to understanding better what goes on in those dark recesses below the surface. And I guess without a water treatment going on, you really need to be compensating if you don't have water treatment with enhancing the level of activity given to any of the other barriers that are going on. So monitoring the water, which is happening much more frequently now, would have also been useful. So this multi-barrier approach is really essential to protecting the water. So really to conclude, microorganisms are everywhere. They're mostly good, but there are a few bad ones, and it's keeping those bad ones away, limiting our exposure to those bad ones is important. Human sewage is the biggest risk, but animal faeces also contains a large number of pathogens. For drinking water, and you need multiple barriers to enable protection, because people drink water that is generally, and everyone drinks the water, you have a large number of people being contaminated. You only need to get it wrong occasionally to have a very large impact, and that impact can be result in deaths. And reducing the pathogenic risk in either recreational waters and source waters really involves in trying to keep those fresh sources of contamination as far away from sources as possible. So, thank you.
Thank you very much. Now, if that wasn't scary enough, we now move into the nitrate pesticide risks as well as the organism risks. So, I welcome Murray Close.
Kia ora. Thanks very much for inviting me along here. I'm going to focus almost entirely on groundwater now, and I've been asked to talk about the risks from nitrates and pesticides to groundwater. So first of all, I'll look at nitrates. Yeah, wrong way. Okay. Okay, just briefly, some people have covered it, but there's basically three aspects in terms of why nitrate is an issue. First of all, there's a drinking water standard. The drinking water standard in terms of just direct ingesting of nitrate is 11.3, and that's primarily just a health risk to us. when we look at the sort of levels that are required for protecting the environment from a eutrophication point of view, the levels are much lower, around 1 to 3 parts per million, will start causing algal blooms, depending on other factors like flows and phosphorus. And then when we look at ecotoxicity of some of these sensitive organisms, some of the macroinvertebrate sort of species, it can be as low as 0.7 parts per million. So the levels that you're looking at depend very much on what the use is and what you're trying to protect. Jan Wright, who's the Parliamentary Commissioner for the Environment, has described nitrate as the elusive pollutant. And the key reason for that is that it's very mobile, it's got a negative charge, and it tends to move with the water. So as the water moves, and we can see, better point at this screen, the nitrate cycle here and interacts with the water. And so as the nitrate accumulates in the profile, it gets carried with the excess drainage water into the groundwater systems. And it moves there very freely. In terms of the sources of nitrate, we've got point sources. They tend to be associated with septic tanks, other waste sort of streams like sewage works, abattoirs, leaking sewage systems, as well as the leaking storage ponds as well. But probably the more difficult way that it gets into our waterways is from diffuse sources. And so this tends to be from grazing animals, especially through the urine, tillage, so that as you till up the soil, you get conversion of the soil organic matter into nitrate, which can then move into the groundwater, and then also fertiliser application. And so this produces lots of nitrate able to get into our groundwater systems. There's very different source loads, I guess, and this is a table taken from Linda Lilburn's report based in Canterbury work, but it'll be fairly similar to the situation here in Hawke's Bay. So up the top there you've got the forests. They tend to give very low levels, but also very low loads. As we come down, viticulture, and then apples in the yellow, and then coming down to some more intensive land uses with some pasture, some crops, and then the red, the dairying and intensive crops. And the interesting thing to me is where the lifestyle blocks end up, and that they're at the bottom of the orange. Concentrations of around nearly 11 ppm can come into can be leached out, and the loads are at about 20 kilograms per hectare per year. So that's quite significant, and it's probably overlooked by a lot of discussions. Now, this is a diagram that Uwe is going to be talking after afternoon tea has put together, and basically looks at the age of a particular groundwater and relates it to the nitrate concentration that's in that groundwater. And you can see very, very clearly that pre-1880, all the groundwater levels of nitrate were very, very low, corresponding to pre-agricultural, basically. And then from 1880 to about 1950s, there was low intensity land use, corresponding to levels of about 2 to 3 parts per million of nitrate. After that, with the intensification, with the irrigation, with fertiliser application, the nitrate levels have shot up, and this, I think, slide really illustrates that very well. If we look at what's it like over New Zealand in terms of nitrates in groundwater, the oranges and the reds indicate fairly high levels, and you can see quite a few, particularly in the Canterbury and Lower South Island. Around Hawke's Bay, doesn't show up very clearly, but there's a mixture of oranges and blues, the blues all being quite low concentrations. And I guess the other factor that I'd like to say with respect to the nitrate, once again, this is a slide from Canterbury, but it's a very similar situation here in the Hawke's Bay, where you've got, whoops, I'm pushing the wrong thing, a lot of input to the groundwater systems from the rivers, and that tends to create a plume of very high quality water at depth. And around here you've got all of the reds and yellows associated with drainage from the land surface sitting on top of this very high quality water associated with the rivers. And that's the same pattern that you'll have here in the Hawke's Bay. Whoops. Okay, how do we get rid of nitrate? Well, the main way to get rid of it permanently is basically from denitrification. And what happens, that's a natural process that happens with the bacteria. If they're in a situation where there's no oxygen, then they'll use nitrate instead of oxygen. And so what you can do, if there's enough carbon, it gets rid of the oxygen, then the bacteria will eat or use up the nitrate. And so if we look at a Pac-Man type of illustration, if we feed it some carbon, gets all excited, chews up the nitrate, and what's given off is N2 gas, which is just the component of the atmosphere. That happens naturally in some situations, particularly when you get into a wetland or in a situation where you've got impeded drainage, high levels of water, therefore you don't have so much oxygen, and denitrification will happen naturally. For a lot of our alluvial gravel aquifers, they're very well oxygenated, and that doesn't happen naturally. One thing we're exploring in our research at the moment is, can we actually make this happen in places we want to for these very fast flowing oxic gravel aquifers? And the idea is that we'll basically, well, one of the ideas we're looking at is to emplace a trench with some wood chips in it and see whether that will actually, as the groundwater flows through this, remove the oxygen, remove the nitrate. Now, Louis Schipper, and that's a photograph from Louis in the right there, has basically installed these in sandy aquifers, fairly slow-moving, homogeneous sandy aquifers, and they work very well. Our challenge is, can we make them work in a fast-flowing, heterogeneous alluvial aquifer? So that's work we've just started last year. Moving on to pesticides. This is just a basic picture. It's fairly old data, but it hasn't changed too much. And it gives the, I guess, the picture of where the pesticides are applied, what are the most intensities. And as we can see, even though pastoral land's about 80%, it uses very, very little pesticides. If we come down to the bottom of the table, horticulture, 1% of the agricultural sort of land, and yet it has quite high loading rates and actually uses 41% of the pesticides there. So the pesticides are a very different sort of contaminant compared to nitrate. They're used extensively, but they're not just a single compound. There's a whole range of pesticides, and they all have very, very different leaching characteristics. The key parameters that we need to look at are the mobility, how fast will they move through a soil, and how long will they last in the environment. And they're basically characterised by the partition coefficient, which is the KOC, and the half-life value. And so because they've got to move through a soil and through a groundwater system, different pesticides will impact groundwaters compared to surface waters. The groundwaters one tends to be the very mobile pesticides that have reasonable persistence. The ones that impact surface waters and then go out into the estuaries tend to be ones that absorb onto clay particles and then move with the clay as they're eroded. So what I did a few years ago is, working with some people from Landcare, was look at a range of pesticides, and what we wanted to know was, are we okay just using the data that we've got from overseas in terms of their mobility and persistence, or do we actually need to do some fresh studies here? Are they going to work in New Zealand the same way? So we looked at 7 sites throughout New Zealand. We applied a range of pesticides with some conservative traces, and we monitored their movement through the profile for between 15 months and 39 months. And so there were sites basically from Northland, right up the top, a couple of sites in Hawke's Bay, which we started with, and then down to the Southland, to give us a very wide range of climatic conditions. Now this is the site, and one of the sites in Hawke's Bay. It was on a Tiawa silt loam just out off Natara Road. And this is the second site, which is in Watson Road, just very close to here, on a Twyford silt loam. And so the sort of results, this is from the suction cup monitoring. We got basically breakthrough curves of the pesticides at different depths through the profile. We also did soil samples. Oops, gone too far. And so at different times, we basically analysed the soil down to a metre and saw how the pesticides were moving through the profile. We put those together with a whole lot of models and estimated the parameters, and basically found that most of the pesticides had very similar mobility and persistence characteristics in New Zealand soils as to what we'd find overseas. There were 4 pesticides, diazinon, which is an insecticide, perchloram, which is a herbicide, prosimidone, fungicide, and triclopyr, which is another herbicide. They had either greater mobility and or persistence than we would have predicted from overseas studies, and so they were much more likely to leach into the groundwater. And in fact, one of those pesticides, prosimidone, we basically found in our pesticide surveys, which I'll come on to next, and that was why we started looking at it here. The other interesting point from that study was that the effect of allophane. Now, alophane's a clay mineral which is found associated with volcanic soils basically, and we found that that could, because it has variable charge, could also affect leaching behaviour of pesticides. So just want to finish up with a brief mention of the national surveys of pesticides that have been carried out in groundwaters over the last 25 years, starting in 1990, basically to see if there was a problem initially, and then to sort of get some idea of whether things were getting better or worse. The first 2 studies in 94 and 90 were detection limits of about 1.1 part per billion. And then for subsequent surveys, it's been about 10 times better than that. So this is a summary table. And what you can see is that over the last 4 studies from 2002 to 2014, there's been between 133 and 160 odd wells that have been sampled. If we look at the higher detection rate, higher detection limit, the rate has been about 10%. If we look at the more sensitive limit, it's been about 20%. So there actually hasn't been a lot of change in terms of the frequency of detecting these pesticides over the last 4 surveys. If we look at a bit more detail about the last survey, most of the pesticides that we detected were herbicides, 86%, and of that, 60% of all the pesticides were triazines. Most of the detections were very low, less than 0.1 part per billion. And there was only one well that had a level greater than the standard, the maximum acceptable value. That was dieldrin, which actually hasn't been used in New Zealand for about the last 50 years. It was used in sheep dip sites all over the place because of basically every farm had a sheep dip site and you had to dip them, and dieldrin was basically used in the 50s and mid to mid 60s. But it's an incredibly persistent pesticide, and so it's still getting there, very low levels, 0.043, but it's quite a toxic chemical, and so the maximum acceptable value is 0.04 parts per billion. The next highest level was a pesticide at 17% of the MAV. So generally speaking, the risk in terms of drinking is very low. As far as Hawke's Bay Regional Council has participated in the last 6 of the surveys, all apart from the first one, and hasn't been detected a pesticide in any of those wells over that whole period. So overall, it's been a fairly consistent frequency of low-level detections, and that's stayed about the same over the last 15 years. Most of the detections are well below the maximum acceptable values. The next survey is due at the end of next year, and we're also talking about incorporating some of these emerging contaminants that Louis is talking about into that survey. And so I think that's me. Thanks very much.
Thanks, gents. We have 5 or so minutes before afternoon tea, so are there any questions on that technical side? Ma'am?
Thank you. Is this on? Yep. I've got a question for John Quinn and Adam Hicks. You're referring to the swimmability standards in your slides as referring to the median of 130 E. coli per 100 mils, which I understand, is that, and I just need to be clarified, is that now the accepted national standard that we're referring to for swimmability targets?
That's what's been proposed in the Clean Water discussion document, which is still under consultation.
So my question is, why are we using it? Or what I understand, and I may be quite incorrect, that we are still supposed to be referring to the 2003 MFE guidelines, or am I incorrect?
Well, I guess the 2 things to refer to for bathing quality is the NOF, the National Policy Freshwater Management 2014 guideline, which had 540 as a 95th percentile. So I mentioned I had that as well. So both there, and the proposal is that they'll both be used, and if you fail one of them, you fail.
So is it? Do I have this wrong? But I'm understanding that these proposed guidelines, these amended proposed guidelines, are actually worse than what were proposed in 2014, where if we compare it back to the 2003 guidelines, the swimmable standards or the acceptable level was based on 6 months of surveillance and 260 CFU per 100 mils as the appropriate guideline, but are we now just defaulting to these new proposed ones?
Well, yeah, we just presented the results into the values that are currently out there, and those are yet to be settled on. Are things being degraded? Well, I mean, until 2014, the bottom line was a median of 1,000 for wading, and what the government's trying to do now is to have a median of 130 and aiming for swimming as being the bottom line. So I think from that perspective, the requirements have got tighter, and it's aiming for a higher target. There has been some extra grades put in the high levels for the high levels of exceedance, things that happen during high flow events typically, and that level has changed a bit. The grading's got more refined, and there is some relaxing of the values at the lower level of those, because it's gone from like 3 grades, essentially A, B and fail, to a 5-grade system with a bit more granularity to it. That's all just what's proposed at the moment.
Yeah, so thanks for answering my question. You're saying that you're defaulting to the new standards which still are not really set in concrete.
Correct.
And are we not supposed to be using the original 2003 MFE guidelines until they've been accepted? Or
Well,
do we just default to these new proposed ones?
You want to have a go? Sure.
Yeah.
I can give up.
Yeah, so none of this was giving us a grade as such. It was more just for context. So just to try to put it into some kind of perspective. Otherwise, you know, some people won't have had the background that you've got. And so it's just trying to give a bit of a context. Are we high? Are we low? Without any concrete tick or cross. Are we failing? Are we passing? Yeah, because that's not yet official. And the NOF is very clear that, or the MPSFM, that new document out of the government, it's very clear that you need to talk to the community, and this is to help that discussion. But the community might actually set guidelines which are quite different to what's proposed at the moment in terms of being more stringent, being of a higher standard. If that helps.
Thanks. Yeah, it does appear that I would have thought that a lot of the community have been quite confused by this, because the 2003 standards were guidelines that were just looking at the high risk period of 6 months of the year, and now it's actually been changed to 12 months of the year.
Sure.
And actually that includes a lot of winter period and winter monitoring when there's actually lower temperatures and higher flows, and that doesn't always relate to increased overall median. So it's just, I think it's been really difficult for the public to get their head around this, and the fact that we're already defaulting to these proposed guidelines makes it even more confusing, but thanks for that anyway.
That's okay. We're actually having a tutorial with our counsellors to try to talk through some of that confusion, because you're right, it is quite confusing. We're confused. So, yeah, good.
Chris? Just wait for the microphone. Chris, can you just wait for the microphone?
Thank you. Dr. Gilpin, I was interested in your set of figures indicating that in your lamb trial you found the presence of Campylobacter, also Clostridium, in quite a high percentage, but that Clostridium is not killed by chlorine. Now, I don't know how common Clostridium is, it's out of my field completely, but if we are relying on chlorine for treating our water here, and Clostridium is in our livestock somewhere, where do we go from here? Because, you know, Clostridium I know is in a lot of animals, but is it here? Do we know where it is? How common is it? So it raises a lot of questions. And ultimately, where do we feed our animals? Because there's a lot of bores in this district that are on an unconfined or in an unconfined aquifer, which will then be potentially contaminated if we have Campylobacter and Clostridium in our livestock using that ground.
Yeah, so Cryptosporidium is what we're referring to. Clostridium is another organism that's present. So the council is actually testing weekly for the presence of protozoa like Cryptosporidium in the water, and I don't believe they've found any. So that's part of the sort of risk assessment of working out what's happened. And so it's an ongoing threat to be aware of, and I guess to be managed, which is what people are doing. And
yeah. But I'd also add to that, Chris, that's why the Hastings District Council is spending a whole lot of money now putting UV plants on the parts of our system that may be at risk from being slightly less confined or where we found young water. So to exactly manage that risk, which is not dealt with by chlorination. Ma'am?
Kia ora. My question is for Dr. Andy Hicks. Dr. Andy, you talked about when I look at the Karamu with all our orange dots, it's troublesome. And what I wanted to say, when you talked about the soft bottom of the Karamu, actually it still has a gravel substrate. What is lacking is velocity because of the diversion of the river near Parkwhai. So we have a main stem Ngaruroro, and then we have the Karamu. Now, the Karamu, as you indicated with your orange dots, without velocity, we can't clean that substrate. I want to ask you one question, and I'm not sure if it's to you or to the engineers, but some of us who have been river users, fishermen, waka ama, rowing clubs, believe that one solution might be to put a permanent mouth closer to the Clive River. Because of the velocity, when the Tutai Kuri and the Ngaruroro are coming around and the mouth has moved toward Waitangi, the Karamu has no velocity or potential to flush out when the weeds are being cut. They're getting their hair cut, they float down, but they can't go anywhere. What do? They gorge and sink and create more substrate sedimentation. Just recently, we have a worm show up in the lower reaches of the Karamu, and it's attached to the Clive pylons on the bridge, and it's moving further toward our marae. It's been seen there. What I'm asking is the velocity, if we can increase that velocity and put the mouth either temporary or full-time or model it so that we get a clean flow out. As you know, we have stormwater, we have urban, we got it all in the Karamu. And so we're looking for solutions to increase that velocity. What would be your suggestions?
There's quite a bit there. That's probably, yeah, I'll talk about the velocity. You're right, I'm probably not, and the council itself isn't probably the only stakeholder or group of people that need to be involved, and I think that'll be something that's talked about during the tank stakeholder process, as well as just the ongoing conversations that the council have with Ko Pātaka and others in the area. The Karamu, the lower parts, there's certainly been that change over time in terms of the flow. Most of the top of the Karamu, though, is above that zone, and so that's naturally soft bottom. But you're absolutely right. There's still gravels underneath that lower river because that's where the Ngaruroro used to flow. It's a big one. So I'm sorry, I might not be able to give you a very satisfying answer today, but it's something that has certainly been talked about before and will be talked about more.
Engineers probably need to look at it.
The engineers have looked at it, so, and Aki, I know you've chatted with Gary and Graham and those guys, so the conversation will continue, and it's all good to have that conversation.
Okay, thank you. One last quick question before afternoon tea.
Thank you, Sarah Gerard. I've got two questions, but I'm probably only allowed one. Perhaps I'll ask the question, since there's two of you on the yes, it's for Brent and Murray. I'm just curious with the trench to help with the idea of removing nitrates. My understanding is anything that, perhaps it's not down into the aquifer, but anything that intrudes down into the groundwater, into the aquifer, may be a form of contamination. And I'm just wondering what Brent's thoughts would be on doing such a treatment, for purification, treatment of nitrates, whether that would be a source of other contaminants into the groundwater.
Murray.
I'm happy to ask that one. Yeah, I mean, that's a real concern. And so at the moment, we're basically just evaluating the idea in terms of will it work. Louise Weaver, who showed some of those Stygofauna, she's also working with some people from NIWA as well and on Stygofauna as well. But we're going to be looking at those, how they're affected by the contamination or the added amount of organic matter we're putting in there. Some of the work that's been done beforehand indicates that we've looked at them below an effluent irrigation application, and in wells just below there, the numbers increase massively as they're processing the organic load, but probably the biodiversity shrinks a bit. And so basically we're going to be monitoring those things, and we're going to make sure that we've got sufficient time or distance to monitor for those effects as the groundwater moves down gradient. So we're going to be looking at both the bacteria that are in the ground, what bacteria are actually doing the denitrification function, and also how are the stygofauna communities being impacted.
Okay, thank you. Look, these gentlemen will be here for the rest of the afternoon. I suggested afternoon tea. If you have further questions, you ask them directly. We are now going to break for afternoon tea, but there is a Datsun ute in the car park, WC 7382, which has its lights on. And also we have a set of car keys if somebody wants to see me over the break, if they're yours. And we'll reconvene in quarter of an hour, and I'll bring you back in when you're ready. Okay, thank you.
I just if anyone wants to see some stygofauna, we've got a couple in jars here of the larger ones that are bigger than a millimetre. So.
Kind of reminds me of the time machine.